molecules like that will form a nematic phase. A nematic phase has long-range orientational order, but no long-range positional order. And in a nematic phase, you can either have tiny distortions in the ordering, which will quickly relax back, or you can have topological defects. And topological defects are systems which are defects which it's very difficult to take out of the system unless you have plus and minus defects annihilating each other. There's one more thing I want to talk about in terms of passive liquid crystals. I want to show you how to write down the continuum equations of motion for passive liquid crystals. So we're still on A here, but we will get a bit further eventually, all right? So it's the last thing in this nematic liquid crystal story, how to write down the continuum equations of motion. Well, what I mean by continuum equations is equations which are like the Navier-Stokes equation, which look at the system not on a molecular level. Oh gosh, let's just get everybody in, shall we? Not on a molecular level, but a much more coarse grain level. So you're all used to the Navier-Stokes equations for a simple fluid. What I want to do is write down the generalizations of these equations for a liquid crystal, for a nematic liquid crystal, which is more complex. Deriving these equations is hard, and I'm not going to do it. So it's really just getting used to the idea of what they look like. And if you ever want to use them, you go away and look them up. And the main thing to take away from the next few slides, which are going to be pretty seriously mathematical, is that we understand these equations. We know how to do them if we want to. And the bit I'm going to concentrate on, which is the most important bit of my lectures, is how you change the equations to go from a passive nematic to an active nematic. And luckily, it's actually rather easy to do that. So these are the continuum equations of liquid crystal hydrodynamics. And the extra thing you need, because this is a complex fluid, is an equation for the order parameter, this tensor order parameter Q. So how does Q behave? Well, first of all, this is the normal advective term, very like you see in the Navier-Stokes equation. Q is advected by the flow field. So if I have a liquid crystal molecule and a flow, obviously it's going to move with the flow. Then you have this extra term here, which I have written out, but I would probably ignore that. It's pretty horrible. But the physics of it is that we have a molecule like that. It's long and thin. So immediately, if there's a shear flow, it's going to have a different flow field at the top and the bottom. And so it's going to turn in any sort of shear flow. So because this is a molecule with this shape, if you have a shear flow, it will turn. And you have to put that in the equations of motion. And you do it in a term which looks like this. You can see here that I've got derivatives in there, which would represent the shear flow. But don't worry too much. Let's just leave it like that for now. And then this term here, that H, is a thing called the molecular field. And it's the derivative of the free energy with respect to the order parameter Q. So what that does, it's a term you put in by hand so that this thing relaxes to the right state. We have here a liquid crystal. It's a nematic liquid crystal. And if you turn off the flow, it's got to do something. And what's it going to do? It's going to relax to the equilibrium state. And how can we describe the equilibrium state? The answer is by the minimum of the Landau-de Gennes free energy. So this thing will relax to the minimum of a Landau-de Gennes free energy. We have to make it do that. And we do that by putting in a molecular field here. That H is dF by dQ, where F is just what we had before, right, when we wrote down the Landau free energy, an expansion in Q, and this thing here, which is just the elastic term in a one elastic constant approximation. So this is really just put in by hand to make it work, to make you get the right state. If you turn off all the velocities, what you want is that free energy to be minimized. Yeah?
So in S, you have rotations of the fluid caused on the particle?
That's the way I'm thinking about it. Yes, essentially.
But you do not have interparticle torque or rotation, right?
You don't worry about interparticle interactions because this is a continuum theory. So essentially they are interacting with each other, but you're putting that in a minute when you do the Navier-Stokes equation as a viscosity term.
Okay, so any kind of interaction so far is in the free energy.
We have to be careful here. Think Navier-Stokes, right? When you're writing down the Navier-Stokes equations, you basically forget about molecular interactions, yeah? And they just come in the viscosity. Same here. So it really is a continuum theory. We don't care about the interactions, we just sort of put them in as the various parameters in there. So it's based on symmetry. That's the important thing rather than actually interactions. Good. Other? Yeah?
In that this assumes it's possible to have shocks in the ordering tensor, or?
Where is my second order derivative?
No, I don't see that. I don't see
Good.
like a Laplacian term. So does it mean that you can have shocks and discontinuities in Q?
I don't think so.
No? Okay.
Yeah? No, sorry. I mean, the second order derivatives would be higher order, all right? So maybe if you drove it in a strange way, you'd have shocks. But no, we don't see anything like that going on here. Remember, this is an expression for Q, not for the velocity. Good. Other questions? All right. Don't worry about the details. It's hard. But that's right. I mean, it's right as in loads of people have used it for 30 years, and it works when you're describing what's going on in, say, a device. So that's the equation for Q. We then have an equation for the velocity u. These are coupled equations. And the way I've written it here, it looks very like the Navier-Stokes equation. This side is the usual inertial terms in Navier-Stokes, and this is the derivative of the stress tensor. And the stress tensor tells me about dissipation in the fluid. I haven't written out E, which maybe I should have done. Normally in Navier-Stokes, that's just the viscosity term, and the viscosity term depends on derivatives of the velocity. So that's just the normal dissipation terms in Navier-Stokes, the normal viscosity term. If that was all that was in there, that would just be the Navier-Stokes equation. But we actually need to add another term in this stress tensor, and it's really horrid. It looks like this. Okay? And the physics behind that term is if I have a molecule like that, or a colloid like that, and it decides to turn, it's going to set up flows. They're called backflows. Okay? And you need to put that in. They're not actually usually very important, but strictly they should be in there. And so you have this term here, which is an elastic term, which accounts for the backflows. Don't worry about it, but just look at this one, which we might see again, I think. This is what that lambda there is, the tumbling parameter. And it's a phenomenological parameter. It's something you have to look at the experiments and see what that parameter is. It's very hard to calculate. I think you can't calculate it. I think that's true to say for a very strongly interacting system. You can for a single colloid, but not for an interacting system. And that tumbling parameter tells me the relative effect of the vorticity, which is trying to turn these particles. Vorticity is the turning of the flows, rotational flows, and the strain, which is trying to align these particles in some way. So let me show you on a movie what I mean by that, yeah?
I just want to remember what we discussed yesterday. But are these equations written for slender particles, or we can just we can assume that they can be spherical too?
These equations are nematic particles, and so they can't be spherical.
Okay.
Okay.
Thank you.
And that's going to come up later because we're going to talk about cells, which certainly look spherical, at least circular to start with, yes. Okay, so this is for a nematic, something with nematic symmetry. Anything else? Let me just show you a picture of what this lambda does. Okay, let's see. If the thing is what you call flow aligning, is that we're going to align? Yeah. When you put a shear flow, what happens is the molecules all organize themselves at some fixed angle to the shear. That's because the extensional flow beats the vorticity, beats the rotational flow. If you change lambda, then what's going to happen is if the vorticity wins, these things are going to do that, and they're going to keep rotating in the flow. Yep. I think the red line is probably where they're going to end up pointing. So, yeah. Okay, so that's two different sorts of behavior you can get from out of these equations if you just put this thing in a simple shear flow. So when I come back to this, which I will, that's the slide I'm going to show you. We have an equation of motion for Q, and we have an equation of motion for U. This is the order parameter, that's the velocity. That's very standard in these sorts of systems where you have structure in your fluid. You need to know how the structure moves in the flow and how the flow itself moves. Okay? And then I've left a space here because I want to put active in there. We're going to see how I change these equations to make them into active equations. And then it is important, I hope that you're going to understand what's going on. But I know this is really hard the first time that you'd seen it. And if it's worrying you, just think of it as a well-known set of equations for a passive liquid crystal. Okay? Any other questions? Do ask questions. Right. Okay, so that's the end of introductions to liquid crystals. Now let's think about low Reynolds number flows. Because, has anyone not heard of a Reynolds number? Good. It would be tough, all right? I will remind you. These things are tiny, these things we're going to look at. Active nematics on a microscopic scale, and so we're pretty much at 0 Reynolds number. So the sort of hydrodynamics we do are going to be low Reynolds number hydrodynamics. And that is going to be very helpful in understanding how to write down the active terms in my equations of motion. So let's look at the low Reynolds number limit. That's the Navier-Stokes equation. I'm terribly sorry. I realize I'm using V for velocity here and U for velocity somewhere else. And I'm afraid I was too lazy to do the slide, but I think that's all right, okay? This is just now the bog standard Navier-Stokes equation for a simple fluid. Forget about the structure. This one here is the inertial term. It tells me how momentum moves through the fluid. This one here is the usual viscosity and is the dissipative term. It tells me how momentum is lost from the fluid. So the way I like to think about it is that if I'm swimming through a swimming pool and I stop moving, you'll glide for a bit, that's this term, and then eventually you will stop, and that's because the momentum is being spread out in the fluid and then lost to the boundaries of the swimming pool. The Reynolds number tells me which of those terms wins, okay? If the Reynolds number is large, the inertial term wins. If the Reynolds number is small, the viscous term wins. And the Reynolds number is a velocity times a length over a viscosity. And for the systems we're looking at, typically the Reynolds number is about 10 to the minus 4. It's tiny. It's 0 for what we care about. Now, I like doing this because it means I can show you pretty pictures. High Reynolds number, large length scales, large velocities. Turbulence, real turbulence, inertial turbulence. Next time you go on a plane, remember that we have these vortices behind the plane which helps lift it up, all right? All right, so these are high Reynolds number flows. Low Reynolds number flows, everything's very calm. This is Volvox, and you can see that no turbulence, things are just flowing very slowly around. All right, and these are the particles of dust in the background fluid which show you where the flows are going. If you've got a microfluidics, the fluid will go very gently down the channel. It will not end up being turbulent at all. You'll get linear flow. And indeed, if you're at very low Reynolds number, trying to mix anything up is really, really difficult. We are going to be talking about active turbulence. It's not the same as inertial turbulence. It can't be the same as inertial turbulence because we're at low Reynolds number. So we've got to find a different explanation for what's going on in these active systems. I mean, the fact that it's called active turbulence, which may have been my fault, I can't remember, ended up getting me into a lot of trouble because people who are real proper turbulence physicists really get upset when you call inertial turbulence proper high Reynolds number turbulence. If you call anything else turbulence, it really upsets them. But I'm using the word in the way somebody in the street would use it, just to mean chaotic flows. Right. So let me show you this, which is a beautiful movie which tells us about fluid dynamics at 0 Reynolds number. If you've seen this movie, you will understand what's going on at low Reynolds numbers. This is a beaker. It's a beaker of glycerol. And the reason it's a high viscosity fluid is that, okay, these length scales are really quite large, but that's okay because the Reynolds number is small because the fluid itself has very high viscosity. So it's the same as having water at very small length scales. And these are blobs of dye which are being put into this beaker. I'm afraid it's a bit slow. That's partly because it is slow anyway, and partly because it's slow because we're tied up to Zoom. But these different colored dyes are put in. And then some graduate students are brought in, okay? This shows you the real length scales, but it is 0 Reynolds number, essentially. And they start mixing it up. And you need to count how many times it's going round. Okay, and I think I can probably, oh no, it's sort of halfway through, so maybe we'll wait. All right, so it's going round. I think it's 7 times it goes round. They have to do it slowly because, of course, if you move it quickly, you don't actually have to do it this slowly, but you have to do it slowly because if you move it quickly, you introduce inertial effects in there. All right, so they're unwinding it now in the other direction. Cute, huh? Okay. At 0 Reynolds number, flows are reversible. Whatever you do, then if you do it backwards, you get back to where you started. That's what makes 0 Reynolds number particularly special. And there are 2 effects of that. This is perhaps the most famous one, so I'll just show you one slide about it, but it's not going to be really very important for us. And that's essentially, we can now answer the question, why do bacteria have these nice wriggly tails? Because if I'm a bacterium and I'm trying to swim at low Reynolds number, which would be the same as me trying to swim in treacle, all right? So I do my swimming stroke and I go somewhere, and then I do the swimming stroke backwards and I go back to exactly where I was before. So this poor little bacterium is using loads of energy to go nowhere. So how do they get round this? Well, to get round it, you have to have a swimming stroke which is different forwards in time and backwards in time. So why might this swimming stroke, when I've got a tail doing this, be different forwards and backwards in time? What is it that's going along that tail? Easy? Come on, what's the thing which goes like that? Waves, thank you. Well done. Yeah, it's a wave, right? And a wave does have a direction in time. So this thing has a wave, and it's okay, it can swim. Similarly, you can also have ones which have a corkscrew-like thing on their back end, because that has a handedness, and having a handedness, if you have a sort of coil, it has a handedness, and so that will give you a direction in time. So that's why these bacteria are these strange shapes. It's so they can move. That's called the scallop theorem, and it's a very famous and very cute theorem. Questions? Just stick your hand up or yell out if you have questions. Okay. However, there's something else about the Stokes equations. The Stokes equations are ones when you get rid of the inertial term and you're just left with the viscous term. So this is just the Stokes equations. It's Navier-Stokes without those inertial terms, which are on the left-hand side. That's linear in the velocity. It's linear in the velocity, which is great, because then if we have a solution, we can add up a lot of different sorts of solution. And there's a whole load of algebra based on this fundamental solution, a thing called the multipole expansion. I'm not going to do, but it's a really cute piece of maths. Okay? This is what happens if I have a point force on my fluid. This is a point force, a delta function. What sort of flow field do I get? If you're more physicist than mathematician, same question. If I pull a very, very tiny colloid through the fluid, what sort of flow field does it set up? And this is the answer. Okay? It's called a Stokeslet, this flow field. A Stokeslet is the flow field set up by a point force. And it sort of makes sense, doesn't it? If you're pulling something through like that, the flow is going to go pretty much like this. This is the mathematical form of the Stokeslet. This is it in vector notation, and that is it in index notation. Sorry, I realize I've switched index as well. I and J are now X, Y, and Z. Okay, so this is index notation. And those are not trivial to prove. You know, one lecture I'd need to get those out, so I'm just going to write them down. But they are done in many fluid mechanics textbooks. Okay, so now I want to worry about an active particle and ask what's happening about the flow field around an active particle. And I'll talk about swimmers because it's sort of easier at this stage. If this particle is active, it's happily swimming away, right? But there's no force on it. There's no net force on it, because where's the net force coming from? By Newton's law, you can't just have a single force, and there's no one sitting there trying to push this active particle around. It's moving itself. So you can never have a single force like that. So what do you have instead? Well, the easiest way it can move is by having a pair of forces. And so what we need is the flow field due to a pair of forces, due to forces like that. It's called a force dipole all right? And for those of you that are mathematically inclined, it's the next ordered term in the multiple expansion. I'm looking at the far flow field around a force distribution, which is my little swimmer. There's no net force because it's active, it's moving itself. So if there's no net force, what's the next possibility? The next possibility is a pair of equal and opposite forces. So let's add up the effect of those two forces. Okay? We know what this one does. It's a little Stokeslet. That's a little Stokeslet in the opposite direction. If you add them up, you do some fairly nasty algebra, finding the flow field here to leading order, adding a Stokeslet at R1 and R2 and saying R1 and R2 are nearly the same, but the difference matters. And what you get, okay, is a flow field like this. It has only an R component, only a component along the radial direction, but that component varies with the polar angle theta. It goes as 1 over r squared. The Stokeslet went as 1 over r. This is the next term in my multiple expansion, so it goes as 1 over r squared. It goes as force times distance between the two forces, so that's like a dipole. Yep. And it looks like this. It looks like this, right? This is my swimmer horizontal there. It's putting fluid out in this direction and pulling it in along the sides. Now, what's the symmetry of that thing? All right. I've written it up there so you can tell me, right? I've written it up here because it's really important, yeah? It's got nematic symmetry. And it has, doesn't it? Because this is a special direction, but that way and that way are the same as each other. So the far flow field around a swimmer, the field a long way away, has nematic symmetry. Now, questions? I know the normal question I get now. Anybody got any questions they want to ask? No? Right. Yeah?
Cosine that appears is just because we are thinking in a dipole in 2D, or in general, it just depends on one angle?
It is, that one's a 3D one, and 2D is, 2D you have to be careful because of log corrections and things. This is 3D, essentially. If that's the direction of the swimmer, any cross-section like that will be the same. Yeah. Yeah?
The topology of the field could be considered a topological defect. I was looking for the shape and thinking that maybe this could be a defect on the velocity field.
I'm looking here at the far flow field. So actually, in terms of the real swimmer, this bit in the middle is never going to happen,
okay,
right?
okay.
Because they're going to be higher order things than just this force. And also defects in a velocity field, because the velocity field is a vector and not one of these headless vectors, are different. So this wouldn't be a topological defect anyway. Yeah, good point, no? Yeah?
So to have this configuration, I don't need that my swimmer have a nematic symmetry. It can be a spherical particle, like a Janus particle, that can do this fluid?
Yes.
Okay.
Yes, that's right. And it's a good point, Nat, that I am, in a way, separating out the symmetry of the flow field, which just comes from the activity of the particle, from the fact that forces act in equal and opposite pairs, yeah, and the symmetry of the particle itself. Yeah, and so when I talk about active nematics, the particles are usually nematic, but actually it's the flow field that's really important. Yeah. Right, usually I get asked, but this particle's swimming, why is it nematic? All right, so let's sort that out. Here I'm looking at the far flow field. Whether it can swim or not is this scallop theorem story, and that depends on the near field. All right? This nematic symmetry is just a far flow field, what's happening a long way away, where I just pick up the leading term from this dipole of forces. Let's just see if it works. These are nice experiments in Ray Goldstein's group in Cambridge. I don't like showing things from Cambridge, but they are beautiful experiments. All right? This is the flow field around E. coli. And these are brilliant experiments because if you look at the flow field around one E. coli, you can imagine it's going to be a complete mess. It's just going to be so many noise and fluctuations, you can't see anything. So they measured the flow fields around loads of these things, I think several hundreds, I'm sorry, I can't quite remember. All right? And then they sort of aligned them all, and this is what they ended up with. And you can see here a very beautiful dipolar flow field out from the ends and in from the sides. That's called, get the right way around, it's a pusher, it's an extensile flow field. By extensile, I mean it's out from the ends and in from the sides. This one's Chlamydomonas, which is this character here that sort of does breaststroke. Yeah? And if you look at the far flow field a long way away, again you see this symmetry, but now it's in from the ends and out from the sides. All right? As long as I'm looking a long way away from that swimmer. So this is called a contractile or puller sort of swimmer. So extensile is arrows this way, contractile is arrows the other way. And I'm going to come back to that a lot, which is why I'm emphasizing this now. You can see this idea of far flow field here, right? This idea of nematic symmetry works, but only if you're a long way away. Here, you get all sorts of junk going on nearer to the swimmer. This story of a dipolar flow field is a far flow field story. Now, interesting, oh, sorry, that was just the extensile and contractile thing. I've forgotten I got a slide there. If you could remember that, all right? All right, so this is an active particle. This is this Volvox thing. But the flow field around a Volvox, that's not right, is it? This is not got this nematic sort of symmetry. All right, with a clue on this picture, why might that be different? Why this particular active particle do you end up with things which look very much like a Stokes slit rather than a dipole? Anyone got any ideas? Yes, sorry? Shout. Yeah, very carefully put on here, right. This thing is quite heavy, so gravity matters. It's bigger than the other swimmers, so gravity matters. So it does have a force on it. It has a net force, which is the force due to gravity, and that wins. And so this thing acts as a Stokes slit because you do have this extra force on it. Right. Okay, so that's A and B done. An introduction to nematic liquid crystals and the Q-tensor, and then we know about low Reynolds number hydrodynamics. Now we can really get going on active systems. Anybody got questions before we do that? Questions welcome? Nope. Okay. There's one there. Yes, hi. That's right.
Brownian motion.
There's no effect of temperature. We're on larger length scales. We're looking at hydrodynamics, not fluctuating hydrodynamics. So temperature really comes in at the order parameter, how aligned, how large the order parameter would be, how aligned the molecules are. Yeah. Very much, this is a continuum. I'm going to be talking continuum. Good point. Yeah. Okay. So now we're going to look at dense active systems. And I realized that actually I should have another one in here, which is just talking about the sort of experiments and the sort of systems that I'm going to be describing. So we learned yesterday, from all the lectures really, what active matter does. And if you think about examples, you can certainly have fabricated active matter like active colloids, but most active matter is biology, right? Active matter is meant to be out of thermodynamic equilibrium. It's always taking energy from its surroundings and using it to do work, and that's what we're doing. We're active particles, pretty big ones, but our cells are active particles, which it's easier to think about. So I'm going to concentrate on dense, wet active matter. So dense, all right, that means I've got lots of these things together. On the whole, I'm going to have lots of them, so they're incompressible. And wet means that I'm using a hydrodynamic theory. I'm thinking of these things as liquids, and indeed as incompressible liquids. That's very different from these flocks, which, if you are going to think about them as liquids, will be compressible liquids, I hope. Right, okay. So let's look at the sort of systems I'm going to think about. So it's nice because I can show you movies. This is how things move around in the cell. So this is a picture of what's going on in your cells the whole time. This is a motor protein. Motor proteins walk along microtubules, so the cell lays down these microtubules, which are long polymers, and then these motor proteins walk along the polymers. And yes, it really is like this. I know this is a computer movie, and certainly the first time I saw it, I said, oh yeah. But it is like this. They have evidence to provide it, and each time you see this movie, it's doing something very slightly different because they've learned something else. Now they've learned that these things go hand over hand in a rotational way. This is a vacuole, which is full of proteins, okay? And the cell walks along the microtubule, lays down microtubules. The motor proteins walk along the microtubules to transport the stuff from one place to another. There is an amazing problem in control theory there. But as far as I know, nobody knows how that's done. But there's a beautiful, beautiful problem. Anyway, perhaps the best-known experimental system in this dense pneumatic active matter is a mixture of microtubules and motor proteins. So these are these microtubules, long polymers, and these are the motor proteins between the long polymers. And these are a bit different because they're two-headed motor proteins, so they hang on to the polymers on either side. And then they walk along the polymers. So these motors walk along these microtubules. Okay? So if I've got two microtubules and the motor's there, and the motors walk in this direction, nothing much is going to happen. But if this one walks that way and that one walks that way, they will push the microtubules relative to each other. So that's why the system is active. These things are being pushed relative to each other by these. About half the motors will be the right way around to do that. And you can see again, you get these dipoles, this dipolar force that you're used to from active systems. And then what you end up with is these sort of bundles of these microtubules which are being pushed around by the motor proteins. And nothing much happens until you add PEG. And what PEG is a depleting agent, so it just makes them denser. And then what you end up with is this rather nice state of active turbulence, okay, where these little white things that you're seeing there are really bundles of microtubules which form, and then the motors sort of pull them apart, and then they move off and form other bundles and so on, and you end up with this state which is turbulent-like, which is active turbulence. What does this look like? Recognize that? Plus a half defect, and this one here, somewhere there's a minus, that one there is a beautiful minus a half, threefold symmetry. Okay, so it's not just random. We're seeing topological defects in this system as well. Oh, I just put this in because it's a nice movie, really. So now I'm going to think about bacteria. So this is system number 2, where you get active turbulence. So this is bacteria. This is another of these molecular motors which is driving things in the cells, a beautiful example of an active system. And this motor is a rotary motor, and it sits here across the cell membrane, and it drives the tail of these bacteria round and round so they can move. And this is, again, an animation, but it's based on many years of research by Richard Berry's group. Each of these things of a different color is a different protein. And somehow in the cell, it manages to create a structure that complicated. And if one of these proteins gets lost, it's able to bring one in and stick it there. It knows how to repair itself. This thing's about probably 30, 40 nanometers long, sitting across the cell membrane, okay? So biology is amazing to be able to create something like that. We can't build something like that, all right? As a physicist, I thought, right, this is active self-assembly, right? It puts everything together. We surely should be able to organize a theory of active self-assembly, which sort of works, at least generically, for all these things. And people have tried, and no one has been very successful. There's some progress for arranging tiles in the right way, but absolutely nothing like this. So I think we maybe have to admit that evolution has done a pretty good job, and it's really annoying not to have one nice physical theory of these things, but we haven't got one yet. Okay, so now if you put loads of bacteria together, this is in 3 dimensions. It's a dense suspension of microswimmers. And again, active turbulence. And then my final example, which we're going to come to in the last lecture, is now, let's get the right word, epithelial cells. Right, so these are squishy cells. They're cells from human bodies, and these are cells which make up maybe about a quarter of the cells in your body. They're skin cells. They're the cells which line the lungs and the stomach. And typically they look like this. This is the cytoplasm in red. These are actin filaments, and that thing's the nucleus. And it's quite hard to see these in vivo in the body, so people tend to take them out of the body and they put them on a Petri dish. So this is a 2-dimensional layer of squishy cells on a Petri dish. On average, these cells are circular, but you can deform them, okay? So you can deform them so they can be pulled so they're longer, and let's see what happens, how these things move. They are getting denser due to cell division, so it's really the beginning of this movie we should look at. Okay? And again, you're getting this random chaotic flows. You're getting something which at least looks like active turbulence. So we've got, oh, I put this one in as well. I won't talk about that one now, but I will later. All right. So we've got 3 different systems there. Do you want to just take 2 minutes, all right, to talk to your neighbors about why, I mean, I've said they're the same, right? I've said this is all active turbulence, but they are different. Just have a discussion with the person next to you. Just think about how these things, how these different systems, the microtubule motor mixture and the swimmers and the cells, how are they different from each other? What might you worry about if you were trying to use the same theory for them? Come on, you're meant to be talking to someone at this point. Okay, okay. So anyone want to tell me a difference between the three systems I might have to worry about? What about someone up there? You going to give it a go?
Suggestions? I don't understand the question. We was talking about the difference about of the three systems, and I think we agreed that is different mechanisms of self-propulsion and alignment. I think is the thing that differ, because in bacterial suspensions you need to induce a flow around in your middle. In the case of cells, you crawl and attach to the substrate or your neighbors. So are different ways to self-propel.
Very sensible. Yeah, thank you. That's not what I would have said, but I think it's better than what I would have said. What about other people? Anyone got any other differences?
How relevant is volume exclusion? So in some of them, they are hardcore bacteria, but in the other one, they are cells that can crawl over each other, right?
Yeah, that's a good point. The way that these particles are joined together, the bacteria are in a background fluid, and the cells are just sort of sitting there on the surface. Anyone got any others?
They have very different shapes. The human cells they can be elongated, but they are spherical, and the microtubules are really long and thin.
Yep, that's probably what I would have said, because we're worrying about deformable cells at the moment, so that's on our minds. Yeah, go on.
Of those, I think only bacteria really move in 3D, because the other ones have to crawl on a surface.
That's true for these particular cells, yeah. And cells in the body definitely move in 3D, but no one's seen active turbulence in the body, but that might be just, it's not a very biological thing, right? That things are not meant to be random in the body. Yeah, so that's right. The bacteria can be in 2D or 3D. Actually, the microtubules can be in 3D, and I'll show you a 3D picture, but those were actually in 2D. Yeah.
And there's differences in time scales and spatial scales?
Not much, actually. Actually, is that wrong? I think that's wrong. Spatial scales are similar. We're sort of 10 to 100 microns with all these things, which is why a continuum theory is great. But the time scales are different because the velocity of the cells is microns per hour, whereas the velocity of the microtubules is micrometers per second. Yeah, that's a good point. Any others? Right. Sorry, say it again. The persistence of the cells and the bacteria? The persistence. Yes. Probably doesn't matter because it's a microscopic thing which would just feed. Oh, yeah, we're very keen on not being the same here. Yeah, go on.
In cells, there might be aging of the bonds, so there's a slowing down in the dynamics. But in bacteria, I don't think there's aging.
Well, they might run out of food. Yeah. Yeah, good, thank you, that's interesting. We're looking at something based on symmetries, so we are looking at a generic theory, and it seems to work fairly well for all these systems. Microscopic details, like the exact nature of the interactions, are not super important. Just like in a fluid, Navier-Stokes works for fluids, for all fluids, and then you have to worry about if you have something like a viscoelastic fluid, yeah. But some details do matter, and I will come to those. Okay, so active matter, why is it interesting? I didn't dare ask you this, because then you'll all be saying, she thinks it's interesting, but I'm not so sure. All right. I used to say that it was interesting to create new types of microengines. Biology is fantastic with these tiny engines, and we can't organize those, but this isn't working at the moment. It's not working because there just isn't enough fabricated active matter to do exciting things with, to do things on an industrial scale. These microtubule motor protein mixtures are great, but they are experimental things in a corner of the lab. Don't use too much of it, right? You don't have a whole big plant. So this is not happening. This is very nice. Non-equilibrium statistical mechanics has been going on for 100 years. It's really hard, and it's got a new impetus by trying to think in terms of these active systems. But what I'm most excited about at the moment is this relation to biology. And it was actually Schrödinger. Schrödinger took some time out from doing quantum mechanics and said that living matter evades the decay to equilibrium. And said, right, yeah, if you're alive, you're out of thermodynamic equilibrium. So using the active matter ideas to look at biology is interesting. How much it's going to tell us about biology, the jury is still out. Biology is horribly complicated. Yeah. Okay, so let's see if the movies can cope with that. Those are those three different systems I'm going to talk about. And I'm going to start by explaining the theory of active turbulence. When I say theory, we understand it, but we do not have a mathematical description of it. So we understand what's going on, but it's not really a theory in the traditional theoretical physics way of thinking about things. Just like with inertial turbulence, we sort of understand what's going on, but we don't have a mathematical theory to explain it. So this is the slide I ended up with earlier. What I want to do is say, what happens to my liquid crystal when it's active? So I've got something, let's think of the swimmers maybe. Swimmers. What extra do I need in there because these things are active? If they were just a load of liquid crystals sitting there, that's one thing, but now they're swimming about, what's different? And the answer is that they're swimming, and so they're creating flows in the surrounding fluid. So what I have to add into my equations is the effect of those extra flows that these things are creating. We know what the flows look like. They're flows with nematic symmetry, which is really why I started from a set of equations which were in terms of this Q tensor, which is a nematic thing. And so any extra thing I put in is going to have nematic symmetry. And it turns out that, in fact, you can do it with a very, very simple additional term in the stress. I have not proved this. I have motivated this. Okay? This is the simplest term you would put in, which has the right symmetry. The active stress, it comes in the stress here. It's equal to zeta, which is just a coefficient which tells you how energetic your active particles are and how many of them there are. And then there's this Q. And it's Q because of the symmetry of the flow fields. The important thing is that this comes in the equations there. It comes in the stress tensor. Okay? So that means, and it comes under a derivative. So any changes in Q, if Q is a constant, it makes absolutely no difference because of this derivative. But any changes in Q are going to give me additional forces acting on my fluid. Okay? Changes in Q give me additional stresses and therefore forces, same thing really, acting on the fluid. And that is the punchline, because this is all I need to explain pretty much everything that's going on in active turbulence. A couple of points. This thing here, this parameter here, zeta. If this is positive, it's an extensile system. If it's negative, it's a contractile system, flows outwards or flows inwards along the long axis of the body. Yep.
Yes, I understand that the stress tensor is aligned with the Q tensor, but is there a reason to assume that the intensity is independent of the magnitude of Q?
It's not really, because that Q has a certain magnitude, and so the magnitude of Q will be in the Q
Yes,
itself.
but it can also depend on non-linearly on
It
Q.
could, yes. So it's a leading order term. You can definitely have quadrupole terms, which is Q cubed, and well, Q squared, actually, yeah, and so on. So anything with the right symmetry can go in there. If you want to just do a symmetry argument,
Yeah.
that has to be the lowest order term.
Yes, okay, so it's a leading term, but you made before a Landau de Gennes expansion, keeping up to Q4. So is it consistent to just keep the first order here?
First order works.
Okay.
Yeah, and actually the only bit of the Landau de Gennes that really matters is the Q squared term, yeah, because the others just stabilize everything. Yeah, well, maybe the Q cube for this one, but yeah. It's a guess, right? It's the first order in a huge expansion, but it works very well. But people have looked at higher order terms, but it's details rather than worrying too much. Yeah, let's go. Another question.
So can we say that this is the condition that an agent needs to improve in the systems to achieve a active turbulence?
Yes. Yes, I'll show you a bit more about this in a minute, how this leads to active turbulence. I haven't really explained that yet, and I'm going to. If people would like to know a little bit more about why this comes out, I actually put it on the problem set, and it's in this paper here, this very famous paper. What is done in this paper is that these little swimmers each have a dipolar flow field, and the paper coarse grains over these dipolar flow fields, tries to add them up to end up with a term like this. As far as I can see, it's a little bit of a fiddle, if you're a mathematician, that particular paper. It's very clever, it gets the right answer, but coarse graining is something it's very, very difficult to do rigorously. And so every time I go through the maths, I go, oh my gosh, that can't be right. All right, I've tried this many times, and I actually think it's wrong. But on the other hand, these people are very clever to get it, because this is the important term that comes out. So if you want to go through and see if you agree with me, this is tricky, but that's how you get this out. Yep.
I don't know if you are going to explain that, but contracting and extensing are related to what? Like pusher and puller? Or
yes.
What?
Extensile is when the flows go outwards from the ends of the thing, and it's a, get the right way around, pusher. Contractile is a puller. Yeah. Okay. Right. So now let's see what happens. I've got two ways of explaining this. I'm going to explain it twice. This is the easy way, and then we're going to have a harder way. So my black ones are the nematic order, right? And I said that this little thing here in a passive liquid crystal, a small bend instability is fine because it will just relax back because of the elastic forces. But this is a place where the order parameter is changing. It's changing its direction. So in an active system, I'm going to have additional stresses, and I'm going to get additional flows. And if you work out the direction of these flows, it turns out to be that way. So what's going to happen is that this distortion is not going to relax back. It's going to just get bigger and bigger, and the nematic order is going to be destroyed. What I'm doing here is a pictorial version of linear stability analysis. Another way of saying that is the nematic state is unstable to activity, and indeed, if it's extensile, it's unstable to a bend distortion. If it's a contractile, it's okay, because the contractile flows would go this way and push it back, but you're still screwed because a contractile system is unstable to a splay distortion. So any sort of pneumatic order is destroyed by these active flows. This is now saying the same thing with slightly more complicated pictures. What I've got here is my tiny little dipoles. So now this is my pneumatic order, but now I've made them dipoles, so I can see that I have flow coming out of the ends and into the sides. And you can see in that nice pneumatic state, all the flows balance and nothing's going to move. But now if there's a tiny perturbation of that state, what's going to happen is that there are going to be more flows here, represented by more arrows, which is trying to push it that way. Here, it will be pushed that way. Here, it will be pushed that way. Here, it will be pushed that way. So a tiny distortion is going to muck you up, and it's going to destroy the pneumatic order. So it's changes in the order parameter give you flows. Linear stability. Oh, I've got a picture. I think I've got the movie. Have I got the movie? Oh, it's just being very slow. That's the problem. Okay. So what I'm showing here, and I will show it again, is this instability in practice in a microtubule motor protein system. This is research from Francesc Segue's group. And the problem is, if you want to see the instability, you have to get the microtubules lined up before you can start. And they don't naturally line up. I mean, it's not really a pneumatic. If you turn off the activity, you don't naturally get a pneumatic phase. You just get everything sticking together in a horrible blob. So the way they've done it is that they put a straw into the middle of this microtubule motor mixture. It's a 2D layer of the microtubule motor mixtures. They stick a straw in the middle and they suck up through the straw. The poor old graduate student has to suck this stuff up through the straw. And because they're sucking it, there are forces and flows. And so what's going to happen is the microtubules will line up, pointing to the straw in the middle. And then they stop sucking, hopefully before the poor old graduate student gets a mouthful of this stuff. And what's going to happen is you'll see the beginning of this instability, and then you actually see a secondary instability in the other direction. So it happens pretty quickly, except it's not going to happen very quickly because of this movie problem. Okay, so I'll play it twice. I think I'm going to have to click that again to make it start. So first of all, you don't see anything because it's lining up. Right, so it's lining up now. And then, oh, it's jumping in a weird way. Here's the beginning of the instability along here. And we're having trouble because of trying. I'd try once more, but I'm happy to show you this afterwards. Not quite sure how many clicks I need to start it. Probably one more. Okay, so there, that's it. That was a nice one then. At least on my screen, I'm not sure about up there. Here you can see the beginning of this instability forming quite nicely. Two different versions here at different time scales. The length scale of the instability, let me just say this because we'll see it again later on. The length scale of the instability is the square root of the elastic constant divided by the activity. At least in the simulations, at least in the continuum theory, it's not at all obvious what this K means in some of the experimental systems. All right, so that's how far we've got. Yep. Sorry,
this instability is a characteristic of pneumatics in general?
Active pneumatics.
Okay.
Not pneumatics.
Active
it's the
pneumatics.
activity that's doing it, but all active pneumatics, right? A passive pneumatic is unconditionally unstable. to activity.
Thanks.
Okay, so that's a summary of where we're up to. Linear stability analysis will not tell you what you get instead, but what you actually get instead is active turbulence. And to see that, what you have to do is you have to actually solve the equations. So this is now this microswimmer picture. This is actually active turbulence of microswimmers in 2D. This is the velocity field when I solve those active pneumatic equations on a computer. If we're lucky. Come on, move. Yep. And what you see is you get these sort of velocity jets. You tend to get, the yellows are regions of high velocity, so you tend to get these velocity jets. And in this active turbulence, you get loads and loads of vorticity. This active turbulent state, if you plot the vorticity, so that's how much the velocity is swirling around, you tend to get these regions of high negative vorticity, which are in red, and ones which are high positive vorticity, which are in blue. And this sort of length scale is maybe about 10 swimmers across. So you get these sort of large-scale vortices formed, and that's very characteristic of the flow fields in active turbulence. All right, so this is the same picture again. This is solving the equations of motion for active turbulence. This is the vorticity in red and blue, and these black lines are the velocity fields. These are simulations, but you get almost exactly the same sort of picture on average from experiments on all these different systems. What I'm doing now is just sort of zooming in on this bit because I want to see the individual particles. So let's zoom in on that bit, and then let's zoom in a bit more. Okay, so here are my velocity lines. These black things here are the individual swimmers. And what you can see is that here, you're definitely getting a gradient in the order of the swimmers, so you get strong forces and stresses. This is a bend instability, and that's creating these flows, which give you these sort of flow jets, and then these bend round to give you the vortices. Yeah? Yep, yep. Oh, sorry, you want to?
So if you have an experiment where you have limited spatial resolution, limited temporal resolution, and even you can only look at a single set position, right? What would you measure to say that you actually are looking at active turbulence?
You can see the velocity. Remember that these things are big. They're not molecular, all right?
Yeah.
They're microns. So you can actually see the velocity field really quite easily for things like PIV.
But you would expect some distribution of the velocity field.
Oh, sure. I mean, this is evolving in time. So this would be a snapshot, but that's fine. You look at a snapshot, and you will get this sort of velocity field. Because the fluctuations are not huge in those length scales. Maybe you're thinking too microscopically.
Yeah, but what is the signature? So I see a velocity field that is complicated. It could be just, I don't know.
Okay. So you'd like me to plot a velocity-velocity correlation function?
For example, yeah.
Fair enough. I carefully took that one out. So velocity-velocity correlation function tends to go like that, where this is because of the vortices, so that you're going up here and you tend to go down there. And actually, the vorticity correlation function also looks very, very similar. Yeah. So people have looked at things like that and do get that out. Yeah.
So these flow fields, yeah, you get the red part is where it's going fast.
No, the red part's where it's going like that, yeah. And the blue is going like that.
But you computed this from the flow fields,
yeah.
Right? So these flow fields, are they faster than the self-propulsion?
Oh, that's a good point. The flow fields sort of are the self-propulsion, okay? Because if you think of and this is one difference between the system, if you think of the bacteria, okay, they're deriving the surrounding fluid, they're driving the surrounding fluid flow. What we're measuring here is going to be the surrounding. I think they're going to be so dense you can't tell the difference. I think that's the right answer to that. They're so dense you can't tell the difference. With the microtubule motor protein system, again, it's the velocity field of the microtubules that you're measuring. But if you have a slightly less dense system, then you'd have to worry. I agree. That's a good question. Okay. Yeah.
So for inertial turbulence, you have a signature, which is energy versus wave number with the power law. Do you have something similar here?
No. I mean, you can sort of see power laws, but they're explainable in a very simple way in terms of the size of the system and things like that. But people have looked a lot, and that was maybe it was bad to call it turbulence because people spent too long looking for these power laws. It's not the same thing, not the same physics.
I think that there are a couple of recent papers of Sankar Rai, I think, where they analyzed the continuum equation. Basically, it was this Toner and 2 Hohenberg-Swiss model, and they found a lot of the signature of inertial turbulence.
I'm surprised, but
like multiscaling, intermittency. They also they have a natural physics from last year, and there is a recent preprint.
I mean, you can certainly see the directed percolation-like crossover to turbulence that you see in active turbulence. You can see in these systems as well. That's one connection. But Toner 2 is not the same, I think.
Yeah. This is absolutely a model equation, so it remains to be seen where it happens. But they push it in a high activity regime. And it's basically Toner and 2 plus some instabilities coming from diffusion and the fourth order term, if I remember well. And that they are in a low Reynolds regime, of course, but they get, I think, different scaling, but all the signature of turbulence.
Right.
I have no idea.
I should have a look at it, because people really tend to say you get scaling and they have it over one decade. But if they see it over lots of decades,
but they applied all the techniques that the people in turbulence use.
Yeah.
And they asked Massimo Cencini, who is an expert, and he was positive about.
Okay. I'll look. We actually did it the same thing. We being Eamon Mohammadi, and we didn't find the same as. But
I think that the trick is going a very active regime, so you need a lot of.
Well,
for them,
if you're very active,
a lot of activity.
The velocity is large, so you may well be pushing, the low Reynolds number limit. Sure. Interesting. Thank you. All right, the jury's still out on that one. Other questions? Okay, there's one more thing we have to do then, because we've got a liquid crystal here. What else are we going to see? That was the active length scale. What else are we going to see? Topological defects, okay? So the most obvious thing to do when you're solving these equations is when you've looked at the velocity field and the vorticity field, you're going to look for topological defects. You don't have long-range pneumatic order, but you do have short-range pneumatic order, and so you can identify topological defects. So we did this, okay? The red dots are +1/2 defects. The blue dots are -1/2 defects. And they do what they're meant to do. They annihilate in plus or minus pair. That one doesn't. I think this one, that one. Gone. Right. But then up here. So what else are they doing? Because the number of defects is pretty much constant. Let me play it again. If I can manage to make it do it. All right. And then you need to look up here, perhaps, later on. there. I think. Okay, see it? Plus and minus defects are popping out. Number of defects is constant. What's happening is that these things are moving around, driven by the flows. If a plus and minus come near each other, they're going to annihilate. But also, they can be created in plus or minus pairs. And that makes sense, right? Because we're active, we're pumping energy into this system. The activity is like having an energy flow through the system. And so there is energy which can locally fight against the elastic distortion, and you can overcome an energy barrier, and you can get these defects popping out. So active turbulence, you have a gas of topological defects which are continually being created and destroyed. And the number of defects gets larger as the activity goes up. So that is very much an active thing. Makes them different from passive liquid crystals. The plus and minus a half defects are pushed around by the flow field, but also they're self-motile. This is the shape of a plus a half defect, yeah? It's pretty obvious that the director field is changing. It has gradients here. It has gradients in direction. Gradients in direction gives you forces. And so this thing, even if it didn't have a background flow, is going to move. And if it's extensile, it moves in this direction. And if it's a contractile one, it moves in that direction. This one, again, has gradients, and it sets up flows, but it doesn't move. And it doesn't move because the flows it sets up have a symmetry, which means there's no preferred direction, and so you're going to end up with it setting up flow fields around it, but not actually moving. Okay, so topological defects are self-motile. This is an experiment. This is an experiment, again, of the microtubule motor protein mixtures, which shows you this actually happening in a real system. What happens is that the forces cause these microtubules to bend like this. And if you look at the bent ones, what's left behind is a minus a half defect with this threefold symmetry, and you see it doesn't move. Whereas the plus a half defect, the one here, is moving towards its head, because this is an extensile system. I'll come back to that. I'm going to actually do that, I think, as a thing. So that's active nematics. Okay, gradients in the order parameter gives you stresses and flows, and then you get these active topological defects. This is generic active nematics, and what we're going to spend some time doing tomorrow is actually asking questions about what extra do you have to put in to look at various, sort of the twiddles on the theory, the extra things you have to put in to look at, say, deformable cells, to worry about the fact that actually a lot of these systems are not nematic if you turn off the activity. They're isotropic. And to worry about what happens if you have surfaces, and to worry about what happens if you have 3 dimensions. But that, I think, is, a good place to stop. And those are a couple of review articles. The first one on active nematics. The second one is nice because it's sort of 5 pages on all the aspects of active matter and just a few references. So it's a really good way to lead into to many topics in active matter physics. Okay, so thanks for listening, and if anybody's got any questions, I'll take the questions.
So thank you very much, Julian. Questions?
Thank you very much. It was very interesting. I was wondering, because I noticed that on one of these topological defects videos that you showed, like the particles like annihilate when they approach, right?
Yeah.
Like, and that behavior looks like a lot of particle-like dynamic, like of each defect behaves like a single particle. And I was wondering, because the video shows like they attract each other, and this is related to the active length scale in some way, or what is that?
Well, the attraction
dynamic
is
of
due
that?
to an essentially elastic interaction, the same as in passive systems. But people have tried very hard to look at this as a theory of particles. Very sensible thing to do. We've tried, other people have tried, and it's really difficult, and I think it's because you have non-equilibrium many-body interactions between these things. It actually turns out to be complicated the way they come together. It's easy if they're facing each other. The minute they're looking in different directions, if you like, it all works in a different way, and then another one up here will make a difference. So, I mean, it really is a many-body system.
Perfect. So that's also this direction what this pair of particles can emerge to. So these to the same kind of active interaction that they have, like because they were attracting, but also they can emerge from a single point of the map. Like
yeah, they can to get them to be formed. You have to have you have to get them over an energy barrier, right? Because two of them have a bit more energy than none, and the flows can do that. They can push the nematogens, they can push the liquid crystal in such a way that they can appear. Okay. Yeah. You can actually have that in passive systems if you have large thermal fluctuations or if you shear them very hard. But then in passive systems, they'll appear, but they'll immediately annihilate because they'll be next to each other. These guys, because the plus halves can move, they move away before they annihilate.
Okay, perfect. Thank you.
So in your continuum model, when you write down the active stress, do you, especially in these dense states, do you need to take into account the change in fluid viscosity due to the particles?
I mean, strictly in a liquid crystal, you might have 5 different viscosities because it depends on exactly which direction you're pulling them relative to the things. That's too complicated. We have enough other things to worry about. The viscosity will change because it's active. It's not a huge effect in that experimentalists don't usually bother about it, but it's something one might want to ask, yeah. We just bung it in as a constant parameter at the moment, yeah.
So in liquids, in fluids, turbulence is a way for dissipating energy. And if we look at active turbulence, that I know it's not turbulence, but what's happening with, for example, entropy production? Is it maximal in that state?
The energy is pumped in at a microscopic level, which makes it very different from inertial turbulence, because each of the little particles is pumping in energy, and then it tends to be lost on the scale of the vortices.
Okay, I see.
So you can, I mean, I haven't done it, but Richard Alert has looked at the energy flow and finds all the terms sort of add up like they're meant to. Yeah.
Thanks.
Okay, some more questions?
During simulations, how do you detect topological defects?
There are ways of doing it basically by looking at a ring around the defect and seeing how much the director field changes around that ring. That will give you the M of the defect. So they're standard plug-in codes in something called image J, which will do it for you.
Okay, thank you.
Talking about turbulence as a way to dissipate energy in the system, there is, across the turbulence, it's often you can see like a lot of scales of turbulence on these classical continuous systems. I was wondering in this active continuum model, you can get like something like that, like different turbulence scales.
No, not really. I mean, this is turbulence. The energy is pumped in at a large length scale and it cascades down. Here, you're pumping it in at the individual particle level, and it cascades sort of up a bit to give you these vortices, but not much more than that. Yeah.
More questions?
Okay.
So let's thank Julia
Coffee.
again.